again on the second day of the workshop. So most of you were present during the first day of the workshop, and we discussed the basics of the project and what we are going to do, what are the deliverables, and how this is going to go, right? We also asked some of you if you have received the kit, and there were questions about printed slit and Foldscopes and this and that. So I can imagine that there are also many more questions that you have about the project, the people who have joined us for the first time today. So feel free to ask questions, feel free to share the grievances or the problems that you are facing, the kind of the way you teach your students. Like some of you prefer a projector, some of you print papers, some of you just teach them directly on a blackboard or a whiteboard. Okay, so moving on, today is the second day of the workshop of monitoring algal bloom, and here are the contents. We are going to talk quickly about the safety features or safety measures that we should all follow while sampling water in the field. And this is something that we will do during the project. Once we are done through the workshop, we will all try to go into the field and collect some water samples and do some cool experiments. I'll also walk you through the protocols of sampling and filtering water. Some of the things that I will talk about, I'll just show you a video, or sometimes I'll do it myself. And then I'll just show you how to look at algae or sample using a Foldscope. Now, one good advantage that you have is that you already have been trained in using a Foldscope. Whatever the Foldscope that you have received, some of you have received a different kind of Foldscope, that's perfectly okay. We don't have a problem with that. So good thing is you all have handled the device, which is a major advantage forward. Then one thing that I strongly encourage is that you all unmute yourself or turn on the video that you of your camera and show us the Foldscope that you have received, if you have it near you, or if you can, then please try to do a quick experiment or just hold it. I mean, this will really help, trust me, when I try to go through the video and show you how to do something. This is going to be, again, some kind of repetition of what you have already done during the workshop with Rafiq, Mo Pandiyarajan, and others. So one quick note is that the kit that you have received, here you all have received a Foldscope. Some of you have assembled it, and others, I'm assuming, like if you have not, please start assembling your Foldscope. And these are the things. Now here you had raised some issues that some of you didn't receive the printed slit, which we will not need in this session today. So we will need it in the next session. So don't worry about it. If you have not received it, I'm talking to Abhishek so that you all get what you didn't get last time. And regarding cardboards and everything, we'll try to manage that. Today is just a demo of Foldscope, and I have some very exciting slides to show you all. So that you will look into the details of human physiology as well.
Iqbal, शायद इरफाना जी का question है.
Yeah. Okay, so please let me know, because on my screen I can't see the chats.
I think she's still connecting. Sorry, go ahead.
Okay. See, yeah. So the first safety measure is that when we go to the field, there are different ways of doing it. So sometimes the teachers that we work with, they go to the field, let's say a water body, a lake, anything where they want to collect water from, they go independently and they bring the water sample to the class and do the experiment with the class, okay? Because it's hard to manage 4 or 5 kids and take them to the field. Sometimes some teachers are much more enthusiastic. So they try to take their kids with them into the field, right? In any case, what we want to make sure is that you don't let the kids roam around in the field because this could be dangerous, right? Depending on the water body that you are targeting, it could be some of the kids may not know how to swim and this and that. So it is super important to take care of the kids when you take them out into the field. Or if you have taught them something, now kids are super curious, they will try to do it by themselves, right? So you tell them strictly that don't go here and there randomly and try to collect water from a place where it could be dangerous, right? Don't go out in bad weather, and please don't drink water from the sources where you collect the water samples, because sometimes some algae can be toxic. Even if the algae is not there, other things could be in the water that are toxic. Now, these are super fundamental, and I'm sure you all teach your students all that, but still it is good to reiterate and before we go forward. If you have gloves, try to wear them, because sometimes we have seen that the water can become super toxic due to algae or some other contaminants, and it can cause skin irritation. Make sure that you wash your hands after you have sampled. The water bodies that don't receive proper sunlight can be really bad and dirty and all. But those are the water bodies where you get fantastic samples. So it's a trade-off. So better to wash your hands before, after at least, when you collect samples, and find a good location so where you can sort of move forward and collect some water, or it's easier to do it. Please don't take unnecessary risk to collect water sample from a place that will cost you. Okay, so please try to be safe, because this is a project where we all want to learn something. We don't want to get injured in collecting water samples and all that. Okay, now this is a protocol that we follow while we collect water sample, or when we go to a field. Now, this is something that you will find on the website. We have not explicitly shared a document with you, but we will send it to you on the WhatsApp group, and we can also email you. The idea is very simple. So when you find a water body and you decide that, well, this is the water body that I'm going to go to every week or every month and collect water. Now, the first thing is try to get a visual understanding of what is there in the water body. Some water bodies are very clean, right? They are visually clean, and some water bodies are super dirty visually. You don't even have to do any tests to find out there is algae. You know that there is algae because some water bodies look just green or just red. There's plenty of things floating on top of the water body. So these are some of the notes that you can take on a printed sheet of paper and sort of collect this information for yourself so that you have a good documentation of what happened visually. Some of the information is not just visual, but also, things like, was the water body smelly? Like, was it smelling of rotten fish or something that might indicate that there is a lot of toxin in the water body due to algae or some other factors. And when you collect so there are some guidelines that we have here. We also have shared pH paper, so you can also measure the pH right there when you collect the sample, if you want to do that. So, you dip the pH and you collect the water sample in a bottle, preferably. If you can see my screen or see my camera here, let me see. Yeah. So
Iqbal,
yeah.
Sorry. I think some people are not able to, they don't have any audio. So please check your systems, your phones, your PCs. I think it's muted because the audio is on, and I think most of us can hear the sound.
Oh, yeah, I mean, I'm not sure why you are not able to hear. But good thing is that this whole process or this video is recorded, so you can always go to YouTube and watch it again. So this is a water bottle that I have, but you all have probably used a water bottle, any mineral water bottle or anything. a standard 1 liter, half liter water bottle is good enough. You can take it with you to the field and just dip it properly and collect a bunch of water out, and just seal it up, bring it back home for experiments. So that's it for collecting water and visually taking a look at what the water body looks like, and we will send this to you. We also have it in different languages. Unfortunately, we don't have it in Hindi right now, but if you think this is super important and your kids would love to have it in Hindi, then we can make it in Hindi as well. So please feel free to tell us what you want. We are here to help. Okay. So Foldscope as a company, so one of the equipment that you have received is this microscope, right? So this one that I have in my hands, this microscope is being made and marketed by a company called Foldscope Instruments. So they have a ton of information online. If you go to their YouTube channel, you can see there are plenty of Foldscope assembly videos on using Foldscope to do many things like looking at coffee, looking at grass, algae, human hair. Rafiq also, posts interesting stuff all the time. So Foldscope has a good repository of information. If you have anything that you are having trouble with, just go online and you will find an answer. They have a good community as well built around this microscope. So that's why we decided to use the microscope, because troubleshooting is easy. Then in order to use a Foldscope, I'm not really very super worried about how to teach you to use a Foldscope, because you all have used a Foldscope. Let me just ask you quickly that we have around 50-ish or 50, how many, around I think 64-ish participants today. So if anyone has not used a Foldscope before, can you just speak up, please? Just let us know that I have not, and it's totally okay. I can teach you right away, or I'm going to do it anyway. Just to see a show of hands, or
आप में से किसी ने भी अगर Foldscope नहीं यूज किया है तो प्लीज बता दीजिए अभी.
Okay, everyone have used it. Cool. Then we just move forward. Okay, so these links will send you over email and also on WhatsApp, so that you can go through these links, and it's just a good recap of what you have learned already. Regarding filtration, let me just go back to my. So there is a workshop 2 video that you will find on our YouTube channel. Let me see. I can't really move this guy. Hold on. Okay, so can all of you see my screen?
Yeah, we see your Zoom screen right now.
Yeah. So this is a video. So we ran a similar workshop with our first cohort in Telangana and other places. So there we have prepared this video, a similar video called Workshop Day 2. There we have extensively explained the filtration protocol. If you collect a bottle of water, how can you filter that water using different tools? You can go through that, and if you have any questions, we can again have another session with you just to walk you through that process. The idea is very simple. You can use different kinds of meshes. You can use cloth as well to filter. And one of the filtration filters.
You just wanted to sort of give a little bit of a background, like why we want to filter.
Okay. Yes. So if you collect a sample from a water body, then the water body, the sample will have a lot of different kinds of algae. It could be algae that are leafy. Let me see if I have. No, I think I'll just show you the. Okay, hold on. Wait, go back. Okay, so so when you collect a water sample, it will have a lot of leafy structures, leafy debris, plant-like, small plant-like, and a lot of scum that you get in your water sample. Now, you don't want it would be really hard to make a sample out of those leafy structures, and it's not that useful because we are looking at algae which bloom and create toxins. So we want to remove that from the sample. So that's why we use different filtration techniques, so that you get rid of all the leafy structures, and eventually what you are left out with are small algae that you can't really see with your eye. So that is there is a reason we have given you a microscope, right? If we wanted to measure algae that you can see with your eyes, then what's the point? So that is the whole reason we are asking you to filter. Next is a demo that I wanted to give you with sample preparation and Foldscope usage. If you guys have any comments, let me know, unmute yourself, or I'll just go forward and show you how to do sampling. So far, as far as I understand, you have not prepared water samples, but some of you may have, because the videos and the pictures that I've seen are all solid samples, not liquid samples. So I'm going to just switch my switch my camera and show you a little bit about how to prepare liquid sample and using the Foldscope. Hold on. So I switched my camera. Can someone pin this screen that I have?
Yeah, I think it's possible to do spotlight on him. I think Kanika or someone.
Okay. So you all can see my screen. So there is something called ring stickers that we have shared with you, which are these things. And you also have glass slides, I'm assuming. So if you, okay, sorry, I got a little distracted. So if you have a sample that is solid, like a hair or some kind of dust or something, then you just place it on the sample and you can tape it with a transparent tape, right? But if you have a liquid sample, it is important to hold the liquid sample in place while you insert the slide into the microscope. Now, one way to do it are these ring stickers, okay? So you can peel them off like that and stick it at the center like that. That's easy. So you have a ring sticker connected to your glass slide. Now, what you can see is that in the ring sticker, there is a tiny plastic flap that you can peel off like that, as you can see. If it is not visible, let me know. I can increase or zoom in on my camera. Let me see. So as you can see, I can peel this flap and there is a cavity that I can expose, right? So at the center of the ring, there is a tiny cavity, right? So this is a water sample, right? It doesn't have any algae in it because it is drinking water. So just to give you a visual, I can use a pipette that you all have and just pipette it up just a little bit, and assuming that this is the filtered sample. So you shake it up because some of the algae may settle down. So you shake it up and take out a little bit of water sample out of it with a pipette, and then insert. Let me see if I'm doing it right. Okay, so you all can see it, right? So I'll just add a little bit of the water sample in that slot, right? And then close it. So if you have more water than the capacity of the ring sticker, then there will be a little bit of spill, but that's okay. Now you have your ring sticker, which has enclosed a tiny volume of the sample. So if you have things moving, let's say when you collect sample from a pond, you'll have plenty of things moving. So they have enough space here to move around. And this also will enclose all the algae, microalgae, and the things that you can't really see, which are green but can't really see with eyes. And then you can insert this in the Foldscope. Now, that's your sample preparation, okay? Regarding Foldscope I mean, visualizing the sample with Foldscope, as you all know, how to use a Foldscope. I'll just walk you through again, and a little, so that you all can remember what you have done once, and I'm assuming you are still doing. So there are multiple components to a Foldscope. So it's a assembled paper microscope. There is a box with which you can shine light from the other side, or you can just point it towards a light source and see with your naked eyes. There is a sample that we need. I just prepared a sample, but there is no point looking at it because it is just water, right? So we won't see anything. It is clean water. So you can pick a sample like this, and you can insert the sample and connect the light source and connect your cell phone. So I recommend that you learn how to use your cell phone properly with the Foldscope, and this will give you a much better hold on taking good images and uploading good images. So these images that you take, it takes some time to become good at it. Now, you all have noticed that I have connected the magnetic coupler on my cell phone. So I couldn't find a better tape. So I just found the paper tape that was there on my desk and connected this magnetic coupler. So it is important to note that this side of the coupler fits to the Foldscope, right, on that side. So you see that it just sticks quickly, easily, right? And I can remove it. But then if you accidentally place it in the other way, then it won't fit. You know the obvious reason: North Pole is North Pole of a magnet, or South Pole repel. Only the opposite poles attract. So magnet always goes in one direction. The other side is this one. It's already, if you have not assembled it, then you can do it. As you can see, it fits properly, but then if you put the other side, it doesn't. So it just bounces back. So I'm going to place it again, so that we can place it, and this fits, right? Okay, so I'll quickly place a sample and show you a demo. And if you have more questions, then we can follow up from there. Okay, regarding placing the sample, this is a little critical. You want to do it very carefully. And one important thing to note here is that you want to make sure that the sample is facing the lens, right? So that's the sample side, and that's just glass. The way I try to do is I just touch it. You can feel the sample, and there is a bump. Here, you see that there are two slots here. They are not obvious, but once you lift up this flap, you see that there is an arrow, right? And you lift up that flap, you see there's another arrow. Let me know if there is a focusing issue with this camera. Sometimes it doesn't work. Okay, so I inserted the slide below the flap, right? So you see, just like that. And now you take it this way and pull the flap and insert it again. It takes some time. So the more you use your Foldscope, the better it becomes in holding slides. Otherwise, early on, there is a bit of tightness that you have to deal with. It doesn't just work all the time, but eventually you get used to the idea. It's good that it is tight because it holds the slide very rigidly, and then you can move the XY stage on top. Now, one thing that I like to do is that I know where the sample is, right? So I can visually place the sample right on top of the lens, so that I don't have to do a lot of maneuvering when I'm connected to the cell phone. And this is like trivial, you just flip it back. So that's where we are. So we have X movement, we have Y, and Z is using this plank, paper-based plank. So you can move it up and down, back and forth. And you can basically, what you are doing essentially with this one is moving the lens away or closer to the sample. So that will give you a different focus, if you are viewing with your eyes, or with the cell phone. Okay, with that, since I'm going to image with cell phone, I want to make sure that my sample is aligned, this is closed, and I just turn the light source on and connect it however I want, because it connects automatically, so you don't have to worry. So I place it there. Now I want to connect my cell phone. So again, I have attached this carefully before the presentation, so I bring it closer and it is already there. So one thing I like to do is place this sample slide holder or anything of this dimension. It could be a pencil box or whatever, that basically makes sure that the cell phone is at the same level. This helps alignment, because if the cell phone is pulling on to the microscope, then sometimes it detaches from the microscope. Okay, with that, I'll just turn my camera on. Yeah, okay, so here we are. I will zoom it in. Can you all see what I'm showing, right? This is a fern rhizome. A fern rhizome is a plant, and it is a cross section of the stem of the plant. So you can see at the center, it has this bright colored cells. They obviously have stained it nicely, so it looks amazing. So you can see that the cells here, oops, I went to a different mode. Okay, have these hexagonal shape or pentagonal shape depending on their location, and they look very beautiful. So like the first time, maybe around 200 years back, when a scientist first made a single lens microscope, his name is Leeuwenhoek. So he looked at it and he was quite amazed. And then there was another scientist who looked at it. Again, they didn't use any sophisticated instrument. They all early on, let's say 100 years back, used a single lens microscope like you all have. And they looked at it and they said, well, this looks like cells, right? In prison, you have cells and rooms and all that. So that's where the name cell came from. So you can see that the cells are packed together tightly in the form of nice hexagons and nice structures. And there's plenty of research that goes on in asking, like, why do they have that specific shape and all that. Okay, now should I open it up to any questions or anything, any issues that you may have faced previously while you were playing around with the Foldscope? I can also zoom in, by the way, with this camera, right? You can all see. And if you have any questions, please ask. आप लोग कोई भी question पूछिए, and we'll try to troubleshoot जो आप लोगों को problems हैं. नहीं तो मेरे पास कुछ interesting slides हैं जो मैंने खुद भी अच्छे से देखा नहीं है, but I thought कि हम सब साथ में मिलके वो experiment करते हैं.
Sir, I am Devendra Kumar speaking from Uchagaon block, UPS Barkon. Sir, I have done an experiment on Foldscope, and in that experiment, I use the Foldscope like a projector.
Oh, you use Foldscope like a projector. Nice.
Yes, sir.
So the advantage of a single lens is that if you flip it and shine light through it, it projects light onto a screen, right?
जी सर, बहुत अच्छी इमेज बनी थी उस पर दीवार पर.
हाँ,
वाइट दीवार थी सर, उस पर बहुत अच्छी इमेज आई थी.
तो आप बताइए, हम बाकी सबको बताइए कि Foldscope को एज अ प्रोजेक्टर कैसे यूज कर सकते हैं.
सर, जो इसका येलो कलर का पार्ट है, उसमें एक होल है सर. येलो कलर के पार्ट में सर, येलो कलर के पार्ट में.
एक सेकंड मैं दिखा देता हूँ, आप बोलते रहिए, मैं डेमो देता हूँ. आप और मैं साथ में मिलकर डेमो करेंगे.
पार्ट है, उसमें एक होल है. ये ऐसे फोल्ड कर देंगे इसको, ऐसे ही. अगर हमारी फोकस, हाँ जी, इसको ऐसे फोल्ड कर देंगे सर, जैसे ओरिजिनल पोजीशन. सर नहीं, ऐसे जैसे इसमें ऑलरेडी लगा हुआ है सर. इसमें इस आई पीस के ऊपर ही ऐसे सेट कर देंगे इसको, जैसे ऑलरेडी रहता है ऐसे ही सर. हाँ सर, उसके बाद में हम अपनी मोबाइल की फ्लैश ऑन करेंगे,
हाँ
फ्लैश,
एक
और
सेकंड
इस फ्लैश,
हाँ फ्लैश ऑन है, फ्लैश ऑन
फ्लैश को कैमरे का यूज नहीं होगा सर उसमें।
अच्छा एक सेकंड रुकिए, मैं थोड़ा मेरा फ्लैश क्यों नहीं ऑन हो रहा है?
क्योंकि
कैमरा
इसमें
ऑन
फ्लैश ब्लॉक है।
I mean, फ्लैश
review
ऑन
the
किया है, बट आई हैव टू टेक maybe I have to go to
आई
video.
थिंक टॉर्च ऑन करने बोल रहे हैं ना,
टॉर्च सर, इसको कैमरा को बंद कर दिए, टॉर्च की जो है
हाँ।
टॉर्च ऑन करने बोल, टॉर्च ऑन
हाँ
करो।
एक सेकंड आई अंडरस्टूड। ओके टॉर्च ऑन है।
टॉर्च को हम उसके बिल्कुल अलाइन करेंगे, ये जो होल है
पीछे की
अलाइन
तरफ
करने,
से, राइट, कैमरा के पीछे की साइड से।
इसको अलाइन करेंगे और सामने जो दीवार है उस पर देखेंगे सर।
हाँ लेकिन यहाँ पे बहुत लाइट है और मुझे ये नहीं पता कि इस रूम का लाइट को कैसे ऑफ करूँ।
सर डार्क होना चाहिए रूम, तब दिख जाएगा वो।
लेकिन
सर
मैं
उस
फिर
पर
भी ट्राई कर रहा हूँ।
सर
Oh,
एक
I
शॉर्ट
can see it. It's very beautiful. Yeah. हाँ,
I
so
actually
मुझे
saw
समझ में
a
आ गया। So
very
basically,
nice post on Microcosmos today. तो Microcosmos, is everybody aware of Microcosmos? यहाँ पे जो वेबसाइट है, तो उसमें भी like basically this is a website जहाँ पे जितने भी Foldscopers है from all over the world and everything, वो अपनी observations यहाँ पे post करते हैं. So this post which just came out 1 or 2 days ago, इसमें kind of similar, this child is using like as a projector और वो अपने किसी white paper पे project कर रहा है, तो वो magnify होके, जैसे आप अनियन की उसमें देखोगे इस लिंक पे, then he's drawing it on the side, जो उसको दिख रहा है माइक्रोस्कोप का, वो उसी के ऊपर ट्रेस कर रहा है व्हाइट पेपर के ऊपर।
हाँ
So
तो
these are
वही
the
मतलब
things, yeah.
आपके आवाज आने लगेगी। ठीक है।
मैंने प्लांट की काइमा का एक मैंने प्रोजेक्ट किया था, उसका वीडियो भी है मेरे पास सर।
You should share it with us.
हाँ आप शेयर करिए हमारे साथ, व्हाट्सएप पे हो या इन जनरल हम हमें ईमेल करिए, हम ट्राई, अच्छा वीडियो होगा तो हम डेफिनेटली पोस्ट करेंगे हमारे वेबसाइट पे।
जी सर, थैंक यू सर।
बहुत अच्छा लगा आपने शेयर किया आपका एक्सपीरियंस, एंड बाकी लोगों से भी आई वुड रिक्वेस्ट प्लीज शेयर व्हाटएवर यू हैव एक्सपीरियंस्ड एनीथिंग, समथिंग सिंपल एज वेल दैट यू हैव सीन और ट्राइड टू यूज फोल्डस्कोप टू डू दैट। ऐसी छोटी छोटी चीजें ही हमें करनी है, एंड बेसिकली स्टूडेंट्स को भी सिखाना है।
हमारे पास में ये जो लाइट वाला इक्विपमेंट है जो आपके हाथ के पास में रखा हुआ है, इसकी लाइट भी हम उस पर कर सकते हैं उनसे।
अच्छा ये वाला ना, ये वाला,
हाँ ये सर, इसमें मैग्नेट भी लगी हुई है।
हाँ
और ये
हाँ, तो आई थिंक दिस इज अ बेटर आइडिया, राइट। आप ये जो है इसको मैं प्रोजेक्ट कर सकता हूँ ऑन दिस प्लेटफॉर्म। अभी दिखेगा नहीं बिकॉज देयर इज टू मच लाइट हियर, राइट।
And the thing that one of the problem with using this is कि ये एक LED kind of source है, like उसको फिर वो diffuse paper इस पे वो लगाना पड़ेगा या तो, तो थोड़ा diffuse light would be better I think.
हाँ,
but, yeah.
diffuse light is better to image actually also with this.
Sir, I want to share my experience here. जो ये लाइट का सोर्स है उसमें जो इंटेंसिटी होती है 2 होती है, 1 and 2 वॉल्यूम होता है। 1 वॉल्यूम पर रखेंगे तो ये थोड़ा डिफ्यूज लाइट ही देगा, हमें इसको अलग से फिल्टर लगाने की जरूरत नहीं है। मैंने तो सर ऐसे ही किया था सर।
हाँ नहीं मतलब आप इसी लाइट सोर्स से अच्छा पिक्चर ले सकते हैं विद अ फोल्डस्कोप एंड कैमरा, बट हमने देखा है कि अगर आप एक डिफ्यूजर लगा दें, एक पतला सा पेपर या ऐसा कुछ जो लाइट का इंटेंसिटी थोड़ा डिम कर दे, जैसे इस रूम में जितने भी लाइट सोर्सेस हैं सबके ऊपर एक हल्का सा ट्रांसलूसेंट पेपर या पैनल लगा हुआ है, तो लाइट डिफ्यूज्ड होती है, तो वो करने से जो क्वालिटी ऑफ पिक्चर्स है वो बेटर होती हैं, हमने देखा है कैमरा में। बट एनीवे आई मीन आप जैसे भी करें, अच्छे पिक्चर्स आए तो इनफ है। मैं अभी बेसिकली एक और स्लाइड लेना चाह रहा हूँ जस्ट टू हैव फन विद इट। मेरे पास एक bunch of slides है and you guys will tell me what you want to see. Okay, so I have a lot of different kinds of slides from Foldscope instruments. So we have, I'll just read it out to you and you can tell me what you want to see. Can everyone see? Is it very blurry?
नहीं, हम read नहीं कर सकते. You will have to.
Okay. So some of it is human skin section, जो हमारा skin है वो, taste bud, skeletal muscle, cardiac muscle, spinal cord, peripheral nerve, kidney section we have. So we all can take a look at kidney section, stomach, intestine, large intestine, breast, ovary, testis, human sperm smear, frog blood smear, duck blood smear, and human blood smear. तो हम basically compare भी कर सकते हैं मेंढक, बतख और इंसान के ब्लड सेल्स के बीच में. और ये काफी इंटरेस्टिंग है. मैं सेल बायोलॉजी में हूँ, अंकिता भी है, आई एम अस्यूमिंग, शायद पहले था. So,
मुझे किडनी देखना है फर्स्ट.
अच्छा, ओके. सो यहाँ पे बहुत इंटरेस्टिंग चीजें हो रही है. माइटोसिस एक इवेंट है जिसमें जो सेल है वो ऐसे डिवाइड होता है. तो माइटोसिस का एक स्लाइड है. क्लीवेज स्टेज ऑफ फ्रॉग एग, जब हम सब लोग एसेंशियली एक सिंगल सेल से बने मतलब एक सिंगल सेल से इवॉल्व हुए हैं. सो वो सिंगल सेल जब डिवाइड होता है मल्टीपल सेल्स में, और फिर एक स्टेज होता है जिसको ब्लास्टुला बोलते हैं, फिर गैस्ट्रुला बोलते हैं. सो ये सब चीजें हैं. सो मे बी आई थिंक अंकिता ने बोला क्या देखना है, सॉरी, किडनी. ओके, अंकिता इज इंटरेस्टेड इन किडनी. आप लोग लिखिए आपको जो जो देखना है. मुझे ह्यूमन स्किन सेक्शन देखना है, तो मैं वो पहले स्टार्ट करता हूँ, फिर हम किडनी पे जाएंगे. ठीक है. Okay, let me try to do it quickly. As you can see, this is human skin. Again, sample किस तरफ है ये देखना बहुत जरूरी है. So it is on this side, so we flip it. We take it up. Again, ये सब solid sample है या sample जो fixed है, किसी चीज में enclosed है and well made है. So you don't have to worry. Liquid samples को handle करना थोड़ा सा मुश्किल है. So you have to get trained in that. So we definitely urge you to start collecting water samples from nearby places. You don't have to necessarily go to a pond. Just go and find a water sample, and you will learn a lot while handling the water sample. Okay, so I have already placed the slide with the sample that I can see. It's a human skin tissue section. So you will see the peripheral layers and the layers below. And I'll just close it up. Okay, I'll place it there. I'll use my holder, and there we go. So this is the first time I'm actually checking this sample, so I also don't know how it will look like. Okay, so I'll zoom it in for you. Can everyone see what I'm showing?
Yeah.
Okay, so here let me just focus a little bit. So as you can see, moving this one, you can focus, right? Because you're moving the lens far away or closer to the sample. Okay, so this part is essentially the top of your skin. So you can see there are plenty of cells, but they are mostly dead cells, so they are differently stained. And as you go down into the skin, the texture and cell and density and everything changes gradually. So I am trying to move deeper into the tissue and see what we find. Oops. Let's see. I'm trying to move it very carefully so that I don't mess up the sample. So as you can see here, oops. I think I'm losing the sample somehow. So these blue dots that you see are the nucleus of the cell. So there are plenty of cells stacked up, and these, I also don't know what these things are. They are very interesting structures. And deeper you have these muscles, and then you have other kinds of muscles which gradually, I don't know why I'm losing focus.
I think once you pressed your phone a bit towards it, it was focusing better.
Oh, okay.
Yeah.
So yeah, here is the deal. देखिए, ये simple सा trick है जो मैं करते ही automatically focus, because phone tilt हो गया था, so वो basically microscope को pull कर रहा था, magnet को भी pull कर रहा था. So now you can see कि deeper into the human skin tissue, you have these big cells. Now there is a difference between these cells and the cells that you saw at the periphery पहले. So ये जो है ये fat cells हैं. हम सब के स्किन में फैट होता है, राइट? सो फैट सेल्स एज अ सर्कुलर गोल गोल मोटे मोटे सेल्स होते हैं, and then as you move up you see epithelial cells, different kinds of cells, and then you have muscles, and यहाँ पे देखिए there is a difference between the texture of skin here and there, right? Okay, so we have a lot of requests, so I'll just go to the next one. Let me just place. Okay, next is what? Kidney?
Yeah.
Okay. Kidney is my favorite because I worked on kidney cells during my PhD. So this is probably a very tiny kidney section. So as you can see, this is the section. It is triangular. So I'm assuming that. So एक interesting fun fact about kidney. Kidney हमारे body में है दो, right? Most of us have two, unless you have congenital defects or something. So most of us have two kidneys. Kidneys are super efficient water pumps. So they filter fluid every day, and they filter at a very high efficiency. Just to give you a number, every day जितना पानी किडनी के थ्रू जाता है उसका वॉल्यूम है 180 लीटर. 180 लीटर हमारे बॉडी में है भी नहीं और हम पीते भी नहीं उतना पानी. So वो it keeps circulating from the blood to the kidney, kidney to the blood again, right? So you can imagine how efficient. And किडनी इतना efficient होता है कि अगर आपका एक किडनी फेल भी हो जाए या you have to get rid of one kidney for some reason, the other kidney can basically back up. वो अभी double efficiency पे fluid pump कर सकता है, and you don't necessarily feel anything. I have a friend जिसके पास एक ही kidney है by birth, and he has no issues, by the way. So phone discharge हो जाएगा actually. Okay, sorry. So I don't know what we are looking at, but let's see. So किडनी के अंदर बहुत सारे ऐसे ट्यूब्स होते हैं जो सिंगल सेल्स से बने हुए, एंड बहुत ज्यादा कैविटी हमें दिखेगा, एम्प्टी स्पॉट्स, वो बेसिकली ट्यूब्स हैं जो जिनके क्रॉस सेक्शन दिख रहा है.
You can already see compared to skin, जैसे skin में जो टॉप लेयर था उसको stratified epithelium बोलते हैं, वो ऐसा एकदम पतला पतला सा होता है. You can see kidney में वो नहीं है.
हाँ. So ये एक इशू है जहाँ पे अगर आपको अच्छा इमेज लेना है, then you have to master the art of holding the phone nicely on your Foldscope, जो मैं अभी भी ढंग से नहीं कर पा रहा हूँ. But you can still see there is a difference between kidney cells and skin cells, and ये जो गैप्स आपको दिख रहे हैं ये ट्यूब्स के क्रॉस सेक्शन है. सो यू कैन इमेजिन कि जो पूरा किडनी है उसमें बहुत सारे ऐसे पानी के ट्यूब्स जा रहे थे, तो मैंने क्रॉस सेक्शन ले लिया तो मुझे सिर्फ होल्स दिख रहे हैं, राइट? सो वो ये सारे ट्यूब्स इंडिपेंडेंटली फ्लूइड फिल्टर करते हैं, वो ट्यूब्स ही पंप्स हैं. और ये मैं किडनी के ऊपर लाइक एकदम बाहर वाले साइड चला गया. यहाँ पे ट्यूब्स का डायमेंशन थोड़ा सा अलग है, बड़े बड़े ट्यूब्स हैं, और या दैट्स अबाउट इट. लेट मी सी, एक बड़ा सा लाइक ट्यूब मुझे मिलेगा क्या जो लेंथ में जा रहा हो? हाँ, ये देखिए. Here you can see some of the tubes which are running parallel to the slice. Can you see? Okay, kidney हो गया. Next क्या है? Should I read the list again? We can go for one more sample. Let me see which one is probably going to be interesting. Human blood smear देख सकते हैं, और नहीं तो हम blastula frog.
Request for taste buds.
Oh, taste buds. Okay. I don't know how taste buds look like. So during the next session, I will request you all to have all the things available next to you so that we all can do the experiment together. And it is necessary because you'll have to make it with me when I show you how to make the spectroscope. Today, actually, we should have told you that maybe you all can also prepare some samples. साथ में experiment करने में अच्छा लग रहा था. Okay. Maybe I need something to add more, you know, of a space here.
If you have a book or something, you can try putting a book.
No, this is not enough space for a book, but I don't know. I'll just hold it like that. Okay. Because I realized it has to be like this. And this is a problem with iPhone because iPhone के कैमरा के just पीछे एक मैग्नेट है, तो वो जो हमारा कपलर है उसको रिपेल करता रहता है, जिसकी वजह से इमेज काफी हेजी होता है. If you have a Samsung phone or some other phone जिसका कैमरा बीच में है, you get a much nicer picture. Okay, so ये basically tongue का cross section है. यहाँ पे बहुत सारे muscles हैं, and I don't know, मैं पहली बार tongue का cross section देख रहा हूँ. बहुत सारे मसल फाइबर्स हैं ये देखिए. So and in between there are something else.
या वो bands की तरह है.
हाँ, so these are muscle bundles. Like ये काफी common है हमारे किसी भी muscular tissue में, like हाथ में and everywhere. So muscle cells क्या करते हैं, वो एक दूसरे के साथ mix up होके they make a big cell and eventually they form a big bundle. Can you see now?
Yeah.
Okay, and ये जो छोटे-छोटे blue मतलब ये overall muscle cell purple है, लेकिन जो blue dots आपको दिख रहे हैं ये nucleus है. तो हमारे body में बहुत सारे types of cells होते हैं. Muscle cells के बहुत सारे nucleus होते हैं, nuclei होते हैं, like plenty of nucleus because वो cells आपस में merge होते रहते हैं, so they make like cells with hundreds of nuclei. ये देखिए, there are so many dots in a single cell. Okay, now quickly, I am very interested in cleavage stage of a frog. So what this is essentially जब like एक single cell होगा frog का, then what happens is it divides into multiple cells. And that division keeps on happening until there is a organism that dramatically comes out of that bunch of cells by self-organization. Okay. Oops. What happened to light source? Oh, yeah, nikal gaya. Light is on, but I can't see anything. So maybe I misplaced my. You see, कभी-कभार क्या होता है कि आपका slide move हो जाता है, so it causes issues. Okay, so you can see this is like the first fertilized egg of the frog, and it has divided into multiple cells. So you can see that there are these cells जो एकदम edges में हैं and कुछ cells बीच में हैं. तो ये जो बीच के cells हैं, ये eventually divide होते होते they will give rise to the frog. और ये exactly, not exactly, but काफी similar processes होते हैं human और mouse और बहुत सारे mammalian development में भी. So as you can see, so you can imagine कि ये पहले एक single cell था जिसको हम egg cell बोलते हैं. So after fertilization, ये egg cell starts dividing and it will form a whole new organism. And so it is extremely potent in terms of many things. Okay, so with that, I think बहुत सारा demo हो गया है. I will stop the demo here and I'll open up the presentation to all of you, and I'll request you to unmute yourself and say something about your experience with Foldscope.
And maybe you want to tell कि अच्छा किसी, like since you guys have been using Foldscope और तुम्हारी workshops वगैरह, you guys have had that. So like what is like, like maybe you can tell us, which will be beneficial for us also to learn, like what are some of the biggest challenges that you guys have been facing so far while you have been using the Foldscope. Maybe some of us want to tell us about that.
हाँ, मतलब my first experience with Foldscope was not all easy. I was struggling with.
It's not all easy. You keep learning.
Right. मतलब I was struggling with the cell phone connection and all that. I didn't really know many things. So if you all have any questions or you can ask something or let us know. I mean, we all are around 73 people here today. So एक दूसरे को help करना is super important. So let us know if you have found something very interesting or you have faced a problem, because I'm sure दूसरे भी यही problem face कर रहे होंगे, उन्होंने बोला नहीं होगा.
I think yeah, let's. I mean, अगर किसी को भी problem है तो please बताइए, क्योंकि फिर otherwise हम assume करेंगे कि सबका Foldscope का usage अच्छा चल रहा है या.
हाँ. अगर कोई प्रॉब्लम नहीं है तो बहुत ही अच्छा है. इधर मैंने बहुत अच्छा समझाया है या बहुत बुरा समझाया है. दोनों में से कोई एक ही होगा. बट
ये पूछ रहे थे हम कि जो फिल्टर, जो सैंपल को फिल्टर करना है, उसके लिए जो फिल्ट्रेशन, जैसे जो पोर साइज है, जैसे कभी कभी एल्गी डिफरेंट साइज की होती है, तो क्या पता अगर उसमें मान लो हमने कोई भी फिल्टर यूज किया, तो अगर हम फिल्टर करेंगे और उसका पोर साइज बहुत छोटा हुआ, एल्गी भी मतलब थोड़ी सी बिग साइज की हुई, तो फिल्ट्रेट हो जाएगी, तो सैंपल में तो नहीं आएगी.
That's a great question. So इसीलिए like हम लोग क्या करते थे, मतलब with the other cohort, like जो तेलंगाना में है या in general, पहले जो हमने शुरू किया था 1 साल पहले, हमने क्या किया था कि multiple filters provide किए थे अलग अलग sizes के, जो इस बार हम नहीं कर पाए because इस बार Foldscope का जो delivery है वो एक दूसरे तरीके से हुआ है, जिसके कारण आई एम नॉट रियली श्योर कि सबके पास क्या क्या फिल्टर्स अभी अवेलेबल हैं. सो यू आर राइट कि अगर हमने लेट्स से कि एक ऐसा नाइलॉन का शीट या कुछ यूज किया जो 100 माइक्रोन का फिल्टर है, तो 100 माइक्रोन से बड़ी वाली जो एल्गी है वो सब अटक जाएंगे उस फिल्टर पेपर में. तो उसका एक सोल्यूशन ये है कि यू कैन स्क्रैप व्हाट द फिल्ट्रेट out, not really the filtrate but what has been captured in the filter paper. आप उसको scrape out कर सकते हैं. जैसे अगर ये मेरा filter paper है and things are stuck here, तो मैं क्या करता हूँ पहले कि I wash it again with another droplet of water, and उससे क्या होता है कि वो पानी का droplet basically takes all the things that are stuck in the filter paper, and then I can scoop out and I can place it again on the slide. So the idea is कि मुझे बस एक अच्छा इमेज चाहिए to identify कि क्या एल्गी प्रेजेंट है ना. That's the whole idea.
दूसरा मैं I also wanted to add कि अगर आप फिल्टर नहीं भी कर रहे हैं, अगर आपको बड़े एल्गी को देखना है, तो अगर आप अपना वाटर सैंपल ऐसे स्टेबल रख देंगे थोड़ा टाइम के लिए, तो जो भारी है वो सेटल हो जाएगा नीचे में और लाइटर वाले ऊपर आ जाएंगे जस्ट ग्रेविटी के उससे। तो उसके बाद आप अपना पाइपेट लेके जब डिप करेंगे तो जो नीचे से लेंगे वो ज्यादा हैवी वाला होगा। तो जैसे बहुत फिलामेंटस एल्गी है, मतलब उसमें बहुत ऐसे ऑलमोस्ट पत्ते की तरह दिख रहा है या बहुत हैवी है, तो वो सब नीचे चला जाएगा। तो वो आप डायरेक्टली ड्रॉपर से ऐसे पिक करके एक स्लाइड बना सकते हैं। आप दूसरा स्लाइड वैसे बना सकते हैं जो सिर्फ टॉप में आपका पानी है उसको लेके एक दूसरा बना सकते हैं, जिससे आपको दो अलग अलग टाइप ऑफ एल्गी का सॉर्ट ऑफ पॉपुलेशन दिखेगा, जो ज्यादा भारी है, जो ज्यादा हल्का है। तो हमेशा भी फिल्टर करने की भी जरूरत नहीं पड़ती है।
और अदर थिंग दैट आई वाज थिंकिंग, बिकॉज as you said this point कि वो settle हो जाते हैं, like again this is not in the frugal dimension, but अगर इनकी labs में centrifuge वगैरह है स्कूल में, I don't know, like स्कूल की labs में से होता होगा? नहीं
No, ma'am, not at all. We don't have any kind of centrifugal in our.
हाँ, मतलब बेस्ट होगा ग्रेविटी बेस्ड सेटल करना।
या जो वैसे पेपर फ्यूज जैसे वो है।
So the best is like if you keep your sample like for let's say 10 minutes, automatically the particles settle there, and then you can also remove the fluid very gently. And there are like clever ways of doing that, and we would be happy to troubleshoot with you. But one thing is super important कि आप लोग collect करिए water sample.
I have seen people using जैसे, हाँ मतलब भाई it will take a little bit of optimization depending कि कौन सा algae वगैरह है, but जैसे coffee filters use कर सकते हैं। So maybe different types of filters जो आपके पास available है आसपास में, you know, just try using all of them and see कि किस उसी sample को if you are using with different kinds of filters, what do you see? तो
कॉफी पेपर इज अ फैंटास्टिक एग्जांपल जहां
और
पे
इवन बहुत पतला जो ऐसे बिल्कुल खराब वाला जो चाय का छन्नी होता है, जो बिल्कुल उसमें मिलता है ना हर एक माल वाला, लाइक उसका बहुत थिन वो होता है, वो बड़े मैश वाला नहीं, जो बिल्कुल पतला प्लास्टिक मैश वाला रहता है। लेकिन प्लीज उसमें अपना या सेम चाय का छन्नी मत यूज करिएगा, लेकिन वो बहुत सस्ता सा शॉप में मिलता है। उसका अगर आप कटिंग कर लें, जो मेटल का नहीं होता प्लास्टिक का होता है, तो वो भी एक बहुत अच्छा फिल्टर है और बहुत पतला फिल्टर है टू सॉर्ट ऑफ फिल्टर आउट एनी बिगर वर्सेस स्मॉलर वन।
हाँ, और जब आप फिल्टर करें, एक चीज मुझे याद आ रहा है, मैं और अंकिता एक एक्सपेरिमेंट कर रहे थे एक बार, तो आप फिल्टर करने के बाद भी जो एल्गी या चीजें हैं वो अक्सर सेटल डाउन हो जाती है। जब आप सैंपल कर रहे होंगे एक बार शेक करिए चीजों को ऐसे करके, नहीं तो ये हो जाएगा कि आपका सारा, या ये देखिए एक
मेरे पास परमानेंटली एल्गी ग्रो होता रहता है। सो अगेन ये बहुत टाइम से वो है, तो आप देख सकते हैं जितना भी भारी वाला है वो नीचे है और जो लाइट, मतलब जो भी लाइट वेटेड है वो सब ऊपर है। लेट मी फाइंड। ओके, सो आपका भी जो भी ड्रॉपर रहेगा, ऑल आई एम सेइंग इज कि आप ऐसे फिर नीचे जाके बिल्कुल धीरे से उसको निकालेंगे, तो देखिए जो जो भारी वाला था वो वाला पोर्शन आ जाएगा आपके पास।
तो 2-3 बार ऐसे सैम्पलिंग करेंगे तो आपको एक ओवरऑल एस्टिमेट मिल जाएगा कि इस पानी में क्या क्या तरीके के एल्गी अवेलेबल है, और उससे जो डॉक्यूमेंटेशन होगा वो बहुत इंपॉर्टेंट होगा। आप अपने स्टूडेंट्स को सिखा सकते हैं, एंड यू विल आल्सो गेट एन अंडरस्टैंडिंग, बिकॉज एल्गी थाउजेंड्स ऑफ स्पीशीज के होते हैं, एंड इट इज नॉट पॉसिबल कि आपके पॉन्ड में या आपके लेक में वो सारे स्पीशीज अवेलेबल हो। आपके पास आपका यूनिक एल्गी सोर्स है, तो इट इज वेरी इंपॉर्टेंट कि आप उसे एक्सप्लोर करिए कि क्या है उसमें। ओके, विद दैट एक चीज, आई मीन इफ देयर आर नो मोर क्वेश्चंस, व्हाट आई कैन डू इज आई कैन गो टू द स्लाइड्स अगेन।
Iqbal, just update that we are already 1 hour up.
Oh, okay. Yeah, we'll wrap it up. So I need 5 more minutes. Okay, so this is a picture of what you might see when you take algae or a sample like that under a Foldscope, and just to see ki, you know, just to share that kaisa dikhta hai. So yahan pe jo at the center that you see is a tiny algae called chlorella, and this algae is the most abundant algae that you will find in your water bodies. It's very hard to like observe the algae because ये बहुत छोटा है। Individual algae एक छोटे गुब्बारे की तरह है, और जो 5 माइक्रॉन्स है ना, बहुत छोटा है। तो मैंने क्या किया, मैंने एक ओवरऑल क्लस्टर, मैंने इसको वेट किया तो ये सेटल डाउन हो गया। मैंने वो जो प्रिसिपिटेट था उसको मैंने पिकअप किया, छोटा सा एक सैंपल बनाया और उसको जूम इन करके दिखाया। सो दिस इज व्हाट कॉजेस मोस्ट ऑफ द ब्लूम इन इंडिया। or the subcontinent. There are other kinds of algae that you can see. These are filamentous algae. There is another algae that you see which has a 2-star dumbbell-shaped chloroplast. This is also quite common, but here you can see this is a nice image that one of our participants has made. He's from Tamil Nadu. And there is another lake picture that we got from California, जहाँ पे you can see कि sometimes पत्थरों पे या ऐसे भी एक algae का एक cluster सा बन जाता है. So you can also pick up some samples from there just to overall understand what the sample or the pond has. Next session में हम लोग spectroscopy क्या होता है वो सीखेंगे and एक spectroscope बनाएंगे on the spot. So I strongly recommend that you come to the session with the tools that we have provided. I mean, Abhishek has provided, and we'll teach you how to make a spectroscope. This week, I'll spend time with Abhishek trying to make sure कि सबके पास सब चीजें पहुंच गई हैं. So don't worry. And here are some of the assignments. आप लोग सब बड़े हैं. There is no need to give you any assignments or take homeworks. I just wanted to make sure that you all go home. You are already at home, sorry. you all start using the Foldscope and check any algae that you can find. And we have an amazing database of freshwater algae called the Algal web. तो आप अगर website में जाएंगे वहाँ पे project page में आपको Algal web बोल के एक सब पेज मिलेगा. तो आप उस पर क्लिक करिए and it is super amazing. हमने एक लाइब्रेरी हम बिल्ड कर रहे हैं जिसमें अल्फाबेटिकल ऑर्डर में फ्रेश वाटर एल्गी के सारे स्पीशीज हैं वहां पे. सो यू क्लिक ऑन अ नेम, यू विल फाइंड अ पिक्चर. तो आपको अगर कोई एक पिक्चर मिले, कोई एक एल्गी मिला एंड यू फाइंड इट इंटरेस्टिंग, बट आपको नहीं पता कि उसका साइंटिफिक नेम क्या है, आप एल्गल वेब में जाके टैली कर सकते हैं या हमें भेज सकते हैं सो दैट वी कैन ऑल्सो हेल्प यू फाइंड दैट. And for the next session, these are the things that we expect you to have. Cardboard, diffraction grating, and printed slit are the things that Abhishek and his team has provided. Scissor, tape, ruler, pen, pencil is something that you'll have to collect or find yourself. So with all that, we'll teach you how to make a spectroscope at home, which is also another fun tool. But the good thing is that we are not buying it, we are making it, right? So usme learning curve bhi better hai. So with all that, I'd like to thank you for being part of this amazing workshop, and we can open the floor for any questions that you may have.
हेलो सर.
जी देवेंद्र जी नमस्कार.
सर नमस्कार, ठीक हैं आप?
बढ़िया,
सर
बताइए.
बहुत सारे हमारे शिक्षक साथी कुछ टेक्निकल इश्यूज या फिर नेटवर्क प्रॉब्लम्स की वजह से ये जुड़ नहीं पाए हैं सेशन से, जो बहुत ही ईगरली जुड़ना चाहते थे लेकिन अभी वो किसी वजह से जुड़ नहीं पाए हैं. तो सर इसका यूट्यूब कुछ लिंक वगैरह इस पे आप शेयर कर सकते हैं?
जी जरूर करेंगे हम लोग. ये सेशन रिकॉर्डेड है तो हम डेफिनेटली इसका एक लिंक है जो आपको शेयर करेंगे.
मैं व्हाट्सएप में शेयर कर दूंगी कल तक. अभी हमारे रात के यहाँ 2 बज रहे हैं तो कल सुबह हम आपको लिंक भेज देंगे.
थिंक लेटर जो सारे लिंक्स वगैरह है स्लाइड्स में, वी कैन जस्ट कंपाइल ऑल ऑफ देम टुगेदर एंड सेंड एज वेल.
सर एक मेरा क्वेश्चन ये भी है कि अगर कोई चाइल्ड हमसे या आपसे या कहीं से भी ये फोल्डस्कोप खरीदना चाहे अपने पर्सनल यूज के लिए, कुछ बच्चे होते हैं कि हम खुद मतलब वो चाहते हैं कि हमारा खुद का अपना हो फोल्डस्कोप ताकि वो ज़्यादा से ज़्यादा उस पर समय बिता पाए. तो ऐसी भी कोई वेबसाइट है या फिर कोई लिंक है या फिर आपके द्वारा कि कोई बच्चा अगर खरीदना चाहे या हमारे स्कूल का हो या कोई और भी हो ताकि वो इसका यूज कर सके?
जरूर, मतलब खरीद सकते हैं और हम कोशिश कर रहे हैं कि हमारा पहले ये कोशिश था कि जो टीचर्स हैं आप लोग और जितने भी टीचर्स हैं उनको एक फोल्डस्कोप मिले और उनके साथ जो 5 या 10 बच्चे जुड़ रहे हैं फॉर दिस प्रोजेक्ट उनको भी मिले. बट इस बार वैसा नहीं हो पाया. बट वी आर गोइंग थ्रू लाइक सम स्टेजेस ऑफ नेगोशिएशन जिससे हम लोग ये भी प्रोवाइड कर सकते हैं.
बट अगर हम नहीं कर पा रहे अभी, वी कैन स्टिल सेंड देम द लिंक्स नो जो इंडिया से इफ यू कैन परचेज. तो अभी भाई फंडिंग का दे माइट हैव टू अरेंज जो भी, बट 300-400 का आई थिंक एक आता है.
So, अगर हम लोग वो नहीं कर पा रहे, एक चीज हम ये कर सकते हैं जैसे कनिका ने बोला कि हम लोग एक लिंक भेज सकते हैं. वो लिंक यूज करके आप इंडिया में ही ये मशीन, ये डिवाइस खरीद सकते हैं. assembled होगा, accessories होगा तो थोड़ा ज्यादा expensive पड़ेगा, या आपका स्कूल अगर इन्वेस्ट करना चाहे, जिस स्कूल में आप हैं, कि बच्चों को एटलीस्ट हैंड्स ऑन ट्रेनिंग देने के लिए कुछ खरीद लें, तो वो भी आप लोग कर सकते हैं.
Yeah, I think like जैसे इंडिया में vendors है जो which are like the Foldscope center, and हाँ अगर that will be actually very nice if you people are able to have some sort of fundraising to give every child a Foldscope. So yeah.
बट मैं ऐड करना चाहती थी, मुझे किसी ने एक कंसेप्ट बताया था जो मुझे बहुत अच्छा लगता है कि फोल्डस्कोप को इधर आपका कोई लाइब्रेरी या स्लैश साइंस लैब टाइप का हो, आप उसमें रख सकते हैं एज अ रोटेटिंग थिंग, जैसे जो लोगों के बीच में शेयर किया जा सकता है. तो अगर आपके पास कोई ऐसा बुक है जिसमें बोला कि अब नेक्स्ट 4 घंटे ये क्लास के बच्चे यूज करेंगे, और आप एज अ टीचर जाके वो वाला फोल्डस्कोप लें, यूज करें, वापस सब कुछ सामान वहीं पे, जैसे लाइब्रेरी की बुक्स यूज करते हैं सेम वे में.
हाँ, आई मीन इट्स इंटरेस्टिंग कॉन्सेप्ट, राइट? अगर एक स्कूल 10 फोल्डस्कोप खरीद ले और उसको एज अ रेंट की तरह दे, जैसे लाइब्रेरी में हम लोग बुक्स. हेलो.
Yeah, I think it's fine. Let's just background.
Okay. Yeah. So next session mein, please, if you have any questions about microscopy, you can bring it up, but please make sure that you have all the things that we have asked you, so that we can do hands-on prototyping together. And yeah, I'm super excited about that. If there are no more questions, I think we can close off today's session.
I think most probably next session भी हम इसी टाइम पे ही रखेंगे.
Yeah, और हम WhatsApp में भी पूछ सकते हैं. आज का attendance is very good, right? So we are about 70 people. So जो बाकी हैं, we will try to make sure कि सबके पास Zoom access हो, and yeah, that is one thing that Devendra ji has mentioned कि क्यों लोग join नहीं कर पाए. So yeah.
Bye everyone.
Thank you. Namaskar.
Hello, ma'am.
Hello. Iqbal, can you
Oh yeah,
stop
yeah,
sharing
sorry.
your thing? Yeah, I can't see who's who.
Let me switch my view to gallery.
सुधीर जी, आपके पास कोई क्वेश्चन है क्या?
Okay.
Okay.
So.
Not we can start winding down.
Yeah. I'm doing that too.